What are cellular organelles? Organelles are specialized structures within the cells that have characteristic shapes and they perform specific functions in cellular growth, maintenance, and reproduction. Each type of organelle has its own set of enzymes that carry out specific reactions and serves as a functional compartment for specific biochemical processes. The numbers and types of organelles vary in different types of cells depending on the function of cell. Although they have different functions, organelles often cooperate to maintain homeostasis. Within cytoplasm, the major organelles and cellular structures include centrosome, cilia and flagella, ribosome, endoplasmic reticulum, Golgi apparatus, lysosome, perioxysome, proteasomes, and mitochondria. The first organelle is centrosome. The centrosome is located near the nucleus. It consists of two components, a pair of centrioles and pericentriolar material. The two centrioles are cylindrical structures, each composed of 9 clusters of 3 microtubules that are arranged in a circular pattern. The pericentriolar material is surrounding the centrioles, which contains hundreds of ring-shaped complexes that are composed of the protein tubulin. During cell division, centrosomes replicate so that succeeding generations of cells have the capacity for cell division. The pericentriolar material of the centrosome contains tubulins that build microtubules in non-dividing cells. This pericentriolar material of the centrosome also forms the mitotic spindle during cell division. Other organelles include cilia and flagella. Cilia and flagella are motile projections of the cell surface. Microtubules are the dominant components of cilia and flagella. Cilia are numerous, short, hair-like projections that extend from the surface of the cell. Each cilium contains a core of 20 microtubules that are surrounded by plasma membrane. Each cilium is anchored to a basal body just below the surface of the plasma membrane. The coordinated movement of many cilia on the surface of a cell causes the steady movement of fluid along the cell's surface. For example, many cells of the respiratory tract have hundreds of cilia that help sweep foreign particles that are trapped in mucus away from the lungs. Flagella are similar in structure to cilia, but they are typically much longer. Flagella usually move an entire cell. The only example of a flagellum in the human body is a sperm cell's tail, which propels the sperm toward the oocyte in the uterine tube. Next organelle is ribosomes. Ribosomes are the sites of protein synthesis. They contain high content of one type of ribonucleic acid, which is ribosomal RNA. Structurally, a ribosome consists of two subunits, one about half the size of the other. The large and small subunits are made separately in the nucleolus, which is a spherical body present inside the nucleus. Once produced, the large and small subunits exit the nucleus separately, then they come together in the cytoplasm. Some ribosomes are attached to the outer surface of the nuclear membrane and to an extensively folded membrane called the endoplasmic reticulum. These attached ribosomes, they synthesize proteins that are destined for specific organelles for insertion in the plasma membrane or for export from the cell. Other ribosomes are free or unattached to other cytoplasmic structures. Free ribosomes synthesize proteins that are used in the cytosol. Ribosomes are also located within mitochondria, where they synthesize mitochondrial proteins. Another organelle is endoplasmic reticulum. The endoplasmic reticulum is a network of membranes in the form of flattened tubules. The endoplasmic reticulum extends from the nuclear envelope, which is a membrane around the nucleus, to which it is connected throughout the cytoplasm. This endoplasmic reticulum is so extensive that it constitutes more than half of the membranous surfaces within the cytoplasm of most cells. Cells contain two distinct forms of endoplasmic reticulum which differ in structure as well as function. One is rough endoplasmic reticulum and other is smooth endoplasmic reticulum. Rough endoplasmic reticulum is continuous with the nuclear membrane and is usually folded into a series of flattened tubules. The outer surface of the rough endoplasmic reticulum is studded with ribosomes, which are the sites of protein synthesis. On the other hand, smooth endoplasmic reticulum extends from the rough endoplasmic reticulum to form a network of membrane tubules. Unlike rough endoplasmic reticulum, smooth endoplasmic reticulum does not have ribosomes on their outer surface. So, smooth endoplasmic reticulum does not synthesize proteins. However, smooth endoplasmic reticulum contains unique enzymes that make it functionally more diverse than rough endoplasmic reticulum. So, rough endoplasmic reticulum synthesizes glycoproteins and phospholipids that are transferred into cellular organelles, inserted into the plasma membrane or secreted during exocytosis. Smooth endoplasmic reticulum synthesizes fatty acids and steroids such as estrogens and testosterone. It inactivates or detoxifies drugs and other potentially harmful substances. It removes the phosphate group from glucose 6-phosphate and stores and releases calcium ions that trigger contraction in muscle cells. Next organelle is Golgi complex. It is found in most eukaryotic cells. It is a part of the endomembrane system in the cytoplasm. It consists of 3 to 20 cisternae, which are small, flattened membranous sacs with bulging edges. The cisternae are often curved, giving the Golgi complex a cup-like shape. Most cells have several Golgi complexes, and Golgi complexes are more extensive in cells that secrete proteins. The cisternae at the opposite ends of a Golgi complex differ from each other in size, shape, and enzymatic activity. The convex entry Orsis phase is a cisterna that faces the rough endoplasmic reticulum, whereas the concave exit or transphase is a cisterna that faces the plasma membrane. Sacs between the entry and exit faces are called medial cisternae. Golgi complex modifies, sorts, packages, and transports proteins that are received from the rough endoplasmic reticulum. Most of the proteins are synthesized by ribosomes that are attached to rough endoplasmic reticulum. These proteins are ultimately transported to other regions of the cell. The first step in the transport pathway is through an organelle called the Golgi complex. Golgi complex also forms secretory vesicles, membrane vesicles, and transport vesicles. Secretory vesicles discharge processed proteins via exocytosis into extracellular fluid. Membrane vesicles ferry new molecules to the plasma membrane, and transport vesicles carry molecules to other organelles such as lysosomes. Next, there is lysosomes. Lysosomes are membrane-enclosed vesicles that are formed from the Golgi complex. They can contain as many as 60 kinds of powerful digestive and hydrolytic enzymes that can break down a wide variety of molecules when lysosomes fuse with vesicles formed during endocytosis. The lysosomal interior has a pH of 5, which is 100 times more acidic than the pH of the cytosol, which is 7. Because lysosomal enzymes work best at an acidic pH, the lysosomal membrane includes active transport pumps that import hydrogen ions. Lysosomes digest substances that enter a cell via endocytosis and transport final products of digestion into cytosol. They carry out autophagy, which is the digestion of worn-out organelles. In this process, the organelle to be digested is enclosed by a membrane which is derived from the endoplasmic reticulum to create a vesicle called an autophagosome. Lysosomes implement autolysis, which is the digestion of an entire cell. Autolysis occurs in some pathological conditions and is also responsible for tissue deterioration that occurs immediately after death. Most lysosomal enzymes act within cells. However, some also operate in extracellular digestion. Another group of organelles are perioxysomes. They are similar in structure to lysosomes, but they are smaller than lysosomes. Perioxysomes are also called microbodies. They contain several oxidases, which are enzymes that can oxidize, that is, remove hydrogen atoms from various organic substances. For instance, amino acids and fatty acids are oxidized in perioxysomes as part of normal metabolism. In addition to this, enzymes in perioxysomes oxidize toxic substances such as alcohol. Due to this reason, perioxysomes are very abundant in the liver, where detoxification of alcohol and other damaging substances occurs. Perioxysomes also contain the enzyme catalase, which decomposes hydrogen peroxide. Hydrogen peroxide is the byproduct of oxidation reactions. Perioxysomes also contain enzymes that destroy superoxide, and without perioxysomes, byproducts of metabolism could accumulate inside a cell, which would result in cellular death. Perioxysomes can self-replicate, and new perioxysomes may form from pre-existing ones by enlarging and dividing. They may also form by a process in which components accumulate at a given site in the cell and then they assemble into a perioxysome. Lysosomes degrade proteins that are delivered to them in vesicles. However, cytosolic proteins also require disposal at certain times in the life of a cell. Therefore, proteasomes are responsible for continuous destruction of unneeded, damaged, or faulty proteins. They are tiny barrel-shaped structures. They consist of four stacked rings of proteins around a central core. These proteasomes are discovered only recently because they are too small to be observed under the light microscope. They contain proteases, which are enzymes that cut proteins into small peptides. Such protein destruction plays a part in negative feedback by halting a pathway once the appropriate response has been achieved. A typical body cell contains many thousands of proteasomes in both the cytosol and the nucleus. Another group of organelles include mitochondria. Mitochondria are referred to as the powerhouses of the cell because they generate most of the energy, that is adenosine triphosphate or ATP, through aerobic respiration, which is oxygen requiring respiration. A cell may have as few as 100 or as many as several thousand mitochondria depending on its activity. Mitochondria are usually located within the cell where oxygen enters the cell or where the ATP is used. These cells are called active cells. Active cells, such as those found in the muscles, liver, and kidneys, which use ATP at a high rate, have a large number of mitochondria. For example, regular exercise can lead to an increase in the number of mitochondria in muscle cells, and this allows muscle cells to function more efficiently. A mitochondria consists of two membranes: inner mitochondrial membrane and outer mitochondrial membrane, with a small fluid-filled space between them. Both of these membranes are similar in structure to the plasma membrane. The inner mitochondrial membrane contains a series of folds called mitochondrial cristae. The central fluid-filled cavity of mitochondria, which is enclosed by the inner mitochondrial membrane, is known as the mitochondrial matrix. The enzymes that catalyze chemical reactions are located on the cristae and in the matrix of the mitochondria. Mitochondria play an important role in apoptosis. Apoptosis is the orderly, genetically programmed death of a cell. In response to stimuli such as large number of destructive free radicals, DNA damage, growth factor deprivation, or lack of oxygen and nutrients, certain chemicals are released from mitochondria following the formation of a pore in the outer mitochondrial membrane. As a result, cell death occurs. Like perioxysomes, mitochondria self-replicate, which is a process that occurs during the time of increased cellular energy demand or before cell division. Synthesis of some of the proteins needed for mitochondrial functions occurs on the ribosomes that are present in the mitochondrial matrix.